Welcome to Sticky Med, learning medicine, stick by stick.
So we all know DNA is like the instruction manual for life, right?
Like the blueprint.
But today,
Yeah.
we're looking beyond the blueprint. We're digging into the actual, machinery, how those instructions get turned into molecules that actually do things in the cell. So let's start right at the beginning. How does the cell decide which gene to turn on or off? That control panel idea. Let's unpack that.
Okay.
So for transcription to even start, you need a signal, a starting line. That's the promoter side, right?
That's it. The promoter is where the main enzyme, RNA polymerase II, and a bunch of other proteins called transcription factors first grab onto the DNA, usually just upstream, before the actual gene sequence.
What makes that spot special? Like, what's the signpost?
It's a specific sequence, chemically speaking. In eukaryotes, you often find these AT-rich regions, lots of adenine and thymine.
Easier to pull the DNA strands apart there.
Precisely, because A and T are only held together by 2 hydrogen bonds, unlike G and C, which have 3. And within that, you look for specific markers like the TATA box and the CAT box. They're like neon signs saying, start here for the polymerase.
And if those signs are broken, like a mutation in the TATA box.
Big problem. The machinery can't bind properly, or it's unstable if it does. You get a huge drop in transcription. The factory just can't get started efficiently.
Okay, so the promoter is the basic on signal. But you mentioned volume dials earlier. It's not just on or off.
Right. That's where enhancers and silencers come in. They add the nuance.
Okay, tell me about those.
Enhancers are specific DNA sequences where activator proteins bind. When they bind, they seriously ramp up the transcription rate, like turning the volume way up.
And silencers.
They do the opposite. They're binding sites for repressor proteins. When those attach, they turn the gene expression way down or even off.
Now here's the bit that always seems kind of wild. These enhancers and silencers, they don't have to be right next to the gene.
Not at all. That's the fascinating part. They can be thousands, even hundreds of thousands of base pairs away, downstream, upstream, sometimes even tucked inside the gene itself in a non-coding bit called an intron.
How does that even work? How does a switch so far away control the promoter?
DNA folding. The DNA isn't just a straight line in the nucleus. It's looped and folded in a really complex but organized way.
Oh, okay.
So when an activator protein binds to a distant enhancer, it can actually cause the DNA to loop around, bringing that enhancer physically close to the promoter.
Like bringing the volume knob right next to the on switch.
Exactly. It creates this transcription initiation complex that really jumpstarts the polymerase. It's all about that 3D structure.
And then there's another layer on top of all this structural stuff. Epigenetics. This feels like context almost. Changes that don't touch the DNA sequence itself.
That's a good way to put it. Epigenetics involves modifications to the DNA or the proteins it's wrapped around that change how genes are expressed. And importantly, these changes can be inherited when cells divide.
Inherited how? Like passed down?
Passed down through mitosis, so daughter cells, remember, sometimes even through meiosis to offspring. But the key is the underlying A, T, C, G sequence hasn't changed at all. It's like adding sticky notes to the instruction manual.
What kind of sticky notes are we talking about?
The 2 big ones are DNA methylation and histone modification.
Okay.
DNA methylation is literally adding a small chemical tag, a methyl group, usually to cytosine bases. This often acts like a do not read sign, silencing the gene. It physically blocks the machinery.
And histones. Those are the proteins DNA wraps around, right?
Yep, the spools for the DNA thread. You can add chemical tags to the histone tails too, things like acetylation. Acetylation tends to loosen the DNA's grip on the histone.
Making it easier to access.
Exactly. It opens up the chromatin structure, making the gene available for transcription. Decetylation tightens it back up. It's a dynamic process, constantly adjusting based on the cell's needs.
Okay, so we've got the signals, promoter, enhancers, epigenetics. The light turns green, transcription starts. Now we're shifting to the factory floor, right? Processing the RNA.
Correct. And in eukaryotes, what comes off the DNA initially is a final product. It's called heterogeneous nuclear RNA, or hnRNA.
Heterogeneous? Why heterogeneous? Sounds messy.
It is messy. It's long, variable, and critically, it contains both the parts that code for protein, the exons, and the parts that don't, the introns, just sort of all strung together. It's unstable too.
So it needs refining before it can go out and do its job.
Absolutely. It has to be processed into mature mRNA before it leaves the nucleus. There are 3 essential steps.
Okay, what's step 1?
5-foot capping. Right at the beginning, the 5-foot end, the cell adds a special protective cap. It's a modified guanine nucleotide called a 7-methylguanosine cap.
Like a little hard hat.
Kind of, and interestingly, this happens while the RNA is still being made. It's co-transcriptional.
Why the cap? Just protection.
Protection is key, yeah.
Yeah.
Protects it from being chewed up by enzymes. But it's also crucial later on. It's the signal the ribosome looks for in the cytoplasm to start translation.
Okay, capped at the front. What about the back end? The 3' end?
That gets a tail, polyadenylation. This happens after the RNA is fully transcribed. It's post-transcriptional. A special enzyme adds a long string of adenine bases, maybe around 200 of them. The poly-A tail.
Is there a signal for where to add the tail?
Yes, a crucial one. Near the 3' end of the transcript, there's a specific sequence, AAUAAA. That's the polyadenylation signal.
And if that signal gets mutated,
Then the tail doesn't get added properly. And without the tail, the RNA is really unstable. It gets degraded very quickly, probably before it even gets out of the nucleus. The tail is vital for stability and also helps with getting the mRNA out.
so capped front, tailed back, what's the third step? Getting rid of the junk.
Exactly. Splicing. This is where the non-coding introns are precisely cut out and the coding exons are stitched back together.
That sounds like it needs to be incredibly accurate.
Oh, it is. It's done by a huge molecular machine called the spliceosome. Any error here, even by one base, could shift the whole reading frame and lead to a completely wrong protein.
So after capping, tailing, and splicing, then we have mature mRNA.
Then you have mature mRNA, ready for export out of the nucleus into the cytoplasm where the ribosomes are waiting.
But even in the cytoplasm, there are more checks, right? The ribosome needs to know exactly where to start reading the message.
Right. Even with the cap, there's another signal right near the start codon. It's called the Kozak sequence in eukaryotes.
Kozak sequence?
This sequence helps the small ribosomal subunit bind correctly and find the actual starting point, the AUG codon. A mutation in the Kozak sequence means translation might start poorly, or not at all, significantly reducing how much protein gets made.
So many layers of control. What happens if the cell makes mRNA but then decides, uh-oh, conditions changed, I don't need this protein right now?
Huh?
Or maybe the mRNA is defective somehow.
Good question. Those mRNAs often get sent to specific zones in the cytoplasm called P-bodies, or processing bodies.
Key bodies, like recycling centers.
Sort of. They have the tools for mRNA disposal enzymes that chop up RNA, X nucleases, enzymes that remove the cap, decapping enzymes, and also microRNAs that can target mRNAs for silencing or destruction.
So they just get destroyed there.
Not always. That's the interesting part. P-bodies can also store mRNAs temporarily.
Store them. Why?
Think about stress response. If the cell suddenly faces starvation, say, it might pause most protein production. It can shunt essential mRNAs into P-bodies to keep them safe.
So when things get better.
It can quickly release those stored mRNAs and restart translation without having to go through the whole transcription and processing pathway again. It's faster. P-bodies are both quality control and a kind of emergency reserve.
Okay, that makes sense. Let's zoom back a bit to the enzyme doing the initial work, RNA polymerase. You mentioned RNA pol-2 does the mRNA. Are there different types?
Yes, definitely. How many types depends on whether you're looking at prokaryotes or eukaryotes.
Okay, prokaryotes first, bacteria and archaea.
Much simpler. Prokaryotes have just one core RNA polymerase, a big multi-subunit complex, but it's responsible for making all types of RNA, mRNA, tRNA, and rRNA. One enzyme does it all.
But eukaryotes, being more complex, specialized.
They divided the labor. Eukaryotes have 3 main RNA polymerases. We number them I, 2, and 3. And conveniently, that order matches the order their products are generally used in making proteins.
Okay, lay them out for us. Pol 1.
RNA pol first is found only in the nucleolus, and its job is to make ribosomal RNA, rRNA. This is the most common type of RNA. You need tons of ribosomes, so pol first is working constantly. It's rampant, as some sources say.
Got it. Pol 2.
RNA pol 2 is the one we've mostly been talking about. It makes the messenger RNA, mRNA, the protein blueprints. It also makes some small regulatory RNAs like microRNAs, mRNA, and small nuclear RNAs, snRNA, involved in splicing. It's the one that finds the promoter and opens up the DNA, the real workhorse for gene expression.
And Pol 3.
RNA pol 3 makes the smaller but essential RNAs, transfer RNA, tRNA, which carries amino acids, and one specific type of ribosomal RNA called 5S rRNA, tiny but vital components.
So 3 specialists. Do they have any shared features? Besides making RNA, obviously.
One really important shared feature, or rather a lack of one, none of the eukaryotic RNA polymerases have a proofreading function.
No spell check, seriously. DNA polymerase proofreads.
Right. DNA polymerase is meticulous about fixing errors during replication, but RNA polymerases, nope. They make mistakes and they don't fix them.
Wow. That really highlights why all that downstream processing and quality control, like the Peabodys, is so critical.
Absolutely. The cell seems to tolerate a certain error rate in transcription and relies on fixing or discarding the RNA later, rather than slowing down the polymerase itself with proofreading.
Now, because these polymerases are so essential, they're also targets, right, for toxins or drugs.
Definitely. Inhibiting them is a powerful strategy. If you want broad inhibition, maybe for lab research, dactynomycin works. It binds to DNA and blocks RNA polymerase movement in both prokaryotes and eukaryotes.
But for medicine, you want specificity. Antibiotics, for example.
Exactly. Look at the rifamycins, like rivampins. These are fantastic antibiotics because they specifically target the prokaryotic RNA polymerase. They gum up the works for bacteria, but leave our eukaryotic polymerases largely alone.
Smart. And then there's a scary example. The death cap mushroom, Amanita phthalates.
Ah, yes. The toxin there is amanitin. And it's deadly specifically because it's a potent inhibitor of our RNA polymerase tubucin.
The mRNA maker.
Precisely. If you shut down Pol2, you shut down the production of almost all new proteins.
And the sources say it causes severe liver damage, hepatotoxicity, and dysentery. Why those tissues in particular?
Because cells in the liver and the lining of your gut are incredibly active metabolically. They have very high rates of protein synthesis and cell turnover. They're constantly making new stuff.
So they feel the effects of protein production shut down first and hardest.
Exactly. When Pol2 stops, these rapidly dividing and working cells are the first to die off on a massive scale. It's a brutal illustration of how central RNA Pol2 is to, well, staying alive.
Okay, so wrapping this up. We've gone from a closed gene to a ready-to-go mRNA molecule. What's the big picture takeaway?
I think it's the sheer number of control points and the layered precision. You've got the large-scale control with enhancers maybe looping DNA over huge distances, down to the nitty-gritty of checking the Kozak sequence or adding that specific 5-foot cap. Every step matters.
It really drives home that it's not just about making the RNA, but making the right RNA in the right amount at the right time and making sure it's, fit for purpose. All those checks and balances.
The capping, the tailing, the splicing, the Peabodys, it's all quality control built into the production line.
So for a final thought, you mentioned enhancers working from far away by looping the DNA.
Yeah, that long-distance regulation is fascinating.
Here's something to ponder then. In that incredibly crowded nucleus, with DNA folded all over the place, how does the cell make sure that an enhancer loops back and activates the correct promoter? How does it avoid accidentally turning on the wrong gene nearby just because the DNA happened to fold that way? That specificity across potentially vast genomic distances, that still feels like a bit of a puzzle, doesn't it?
It absolutely does. Maintaining that fidelity, avoiding that kind of crosstalk in such a dynamic 3D environment. That's definitely one of the frontiers. How that nonlinear genome geometry works with such precision is still something we're figuring out.